Okay, so bioinformatics is just quite new branch of the in biology. Basically, we integrate both two branches of the different field together, okay, to analyze the informations of the biology. So basically involve the biology and also the IT, the information technology, okay, software development. So first we need to define bioinformatics. Sorry, bioinformatics. So this bioinformatics basically it's a system for us to collect the data, to process, to analyze, and very important, to search information. So allow us to search for a particular informations. Okay, so now we need to outline the role of bioinformatics following the sequencing of the genome, such as why we look at the humans and parasites. Okay, genome for what? Okay, so how we can use this bioinformatics system. Not only this, we have a lot of the system or a lot of software that we can use to help us in terms of the study of the bioinformatics. And number three, we're going to discuss the molecular evidence that review the similarity between the closely related organism with reference to the mitochondrial DNA and the protein sequence data. So how we do the compare, how we actually look at the similarity, how we can conduct, to conduct this bioinformatics using bioinformatic tools to give us what we call the family tree. Are you clear? So how closely related we and the chimpanzee, we and the tiger. So we can use this bioinformatics tools to help us. Okay, so we started with the introductions to bioinformatics first. So basically bioinformatics is defined as it's a collections that we can process or analyze the biological information and data using computer software. Okay, so very important. The whole world doing the sequencing. We don't want to repeat that someone have been done. So we want to collect the data. It's a database basically. Bioinformatics system is a database for us to collect, to process, to analyze the data and for the searching. For example, China have done the COVID-19 sequence, right? Malaysia have done our sequence. Okay, we know that it's alpha mutation, beta mutation, delta, for example, right? India have done, US, European country have done. So how we can compare? We need to have a database. So this bioinformatics actually serve as a database to collect. And we can process. How we process? We can do the compare. We can analyze. Okay, how come we know that, oh, this is delta? How come we know that this is alpha mutation? How we know? So based on the database. And very important, they must be allowed us to access and search. Are you clear? It's not like a private database. It's an open database. You, me, anyone can go and search. And then we can actually access, search, and we can do the compare. So where we compare here, how we collect, what kind of biological sample or data that we actually collect. Okay, so we actually collect gene sequence, genome sequence, a complete genome. Okay, amino acid sequence. As long as we can have the sequence, then we can. And also we can have the protein structure. Means that we do have a sequence of amino acid, we can do the prediction how the proteins can fold or polypeptide fold in the precise three-dimensional shape, then we can have the protein database. Okay. So what we apply, how we apply this bioinformatics system, we can apply in the medical field. For example, in terms of molecular medicines. Okay. Like for example, just now we look at a microarray. Okay, microarray tell me that this gene expressed. So we're going to search a suitable drug to be used. Clear enough? Gene therapy. We're going to learn gene therapy the next subtopic next week. Okay, pharmaceutical and biotech industry. We're going to have the protein structure predictions. For example, some proteins that we want to see how it fold. After it fold already, what kind of the ligands we can use to inhibit that particular protein. Just now we know that, oh, protein A expressed highly in the cancerous cells, but we want to inhibit that particular protein. But so many drugs, how are we going to do? How will we know? So basically, we can use the software to predict the binding. We're going to learn this next week, okay? Particularly in drug development. For example, in COVID-19, right? We want to do the vaccinations. So how we actually come out with this vaccine? How we know which part we're going to target? Why we have the mRNA vaccine? Correct or not? Okay? How we use the liposome to encapsulate. So those drug developments, we need the gene sequencing data. The whole world share the data, then we'll be able to develop the drug vaccine in this process. We can do the comparative study. We compare the genomic study. For example, most of the times we want to carry out the experiment, we cannot straight away do it. Let's say, for example, drug. Oh, we developed the drug to treat COVID. But first thing you need to have the model to study. Cannot. Okay, we study on the rat, we study on the mice, monkey, chimpanzee. Cannot. Why we don't study on chicken? Why we don't study on, let's say, for example, in tiger? Because genomic studies so that we are closely related to chimpanzee. So chimpanzee can be the one that we use for the preclinical study. Okay, so we also can compare the analysis of mitochondrial DNA. Okay, compare with between two different organisms, how far we share the ancestor. Okay, how far? Basically, we want to know. Okay, from humans. Okay, evolution take place, right? Okay, sorry, evolution take place from the primate. So how far we share the common ancestor? We can do the phylogenetic or evolutionary biology, okay? So that we can construct, okay, this evolutionary tree as well as the phylogenetic analysis. So these four, we're going to focus where later we're going to look at each kind of the tools that we can use in the bioinformatics. So first, we can have the gene identification and sequence analysis. Okay, one of these we call it the BLAST. BLAST stands for Basic Local Alignment Search Tool. The BLAST, I give you the URLs. You can actually go and search it. Okay, just try and play around with it. So BLAST find the region of similarity between the biological sequence. The program compare the nucleotide or protein sequence to sequence database and calculate the statistical significance. How much, how similar the two gene is. Let's say, for example, you go to for expedition. Hey, you saw an organism. Hey, what's special? I never seen it before. So you take the specimen back, you do the sequencing, the genome sequencing, and you want to search and know, hey, closely related to which organism. So we have the gene, we blast it. Because thousands, even millions of the nucleotide sequence, how nucleotides, right? How we compare AA, TT, CG, look at one by one. Human tends to make error, but we can use a computer software here, BLAST, to help us. Okay. And after I actually done the analysis already, it doesn't match with any organism. Is it a new organism? Then I can conduct the phylogenetic analysis to see how closely the new species that I have with the existing species. When the evolution take place, when is the speciation actually take place? Can I see that? Okay, and I can have a nucleotide database. Okay, to store all these nucleotides. It's open access, means everyone can actually get the access. And we can actually do what? In this case, when you have a nucleotide database already, I can compare humans with the rat, humans with the mice, and then we can use the rat and mice to study, okay, the disease. We can have the protein database also. We have two UniProt and the RCSB. So both of them can give us the three-dimensional. Next week, I'm going to show you the structural three-dimensional, how we can actually rotate the protein and see where is the binding site, or rotate, okay, the enzyme to see where is the active site, allosteric site, okay, whether we can use a inhibitor to inhibit them or not, okay. And we can have the genome database. Genome database is the joint scientific project between the European Bioinformatics System and the Wellcome Trust Sanger institutions. Where we do, they actually to provide a centralized resource for the geneticists, molecular biologists, and other researchers study the genome of our own species, human, and compare with other vertebrates and some model organism. Are you clear? The ensemble here. And finally, we do have another one called molecular docking. Molecular docking, actually, SwissDoc is not a free software. I may not be able to show you guys, okay? But this SwissDoc actually shows us what? For example, we do have the protein that causes the disease. So we want to find a suitable drugs. But in this world, we have millions, even trillions of the drugs. Which one's suitable? If you want to do experiment and one by one, it's going to cost us a lot. It's going to cost, times, okay? We'll use up a lot of time. But what we can do, we can use computer system predict. Okay, we can use, right? We can actually insert our protein inside, and then we can put in all the possible drugs, and we can predict the binding effect. Oh, binding very well, inhibitions very well. Then maybe this is a suitable drug. Then we carry out the experiment to prove it. Okay, so with this, I'm going to stop it here. Okay, next week only I will go into the all these tools. Okay, so next week. Okay, so far, do you guys have any questions regarding to the bioinformatics or not?